Okay. Okay, hi everyone. So I'm going to be talking to you this morning a little bit about the project that I've been working on this past couple of weeks, which is developing BearID for brown bears using camera trap imagery. So I just wanted to give you a little bit of background about our study area. So we are based at the southern end of the Great Bear Rainforest, which is this large tract of what's called contiguous, so it means connected, temperate rainforest. It's actually one of the largest remaining in the world, and it stretches from about mid-Vancouver Island all the way up to the border with Alaska and the Alaskan Panhandle. And this is what it looks like. Wow. So yeah, it's as I say, temperate rainforest ecosystem that's carved by these glacial fjords called inlets, and so we can only really access these areas by float plane or by boats. And so bears use their kind of home ranges within these areas. They focus a lot of their activity on estuaries, on intertidal zones. They sometimes use ridgelines to move, but a lot of the activity is centered around river systems and estuaries. So that's where we concentrate most of our studies. So we use cameras and direct observations in one particular river system called the Glendale River. To study bears. We have a number of different research projects going on, some studies looking at social communication in bears, others looking at human impact. But we also have a large long-term kind of more general monitoring of them, where we monitor individual bears over time. And part of that work involves recognizing individuals. So we, over the years, this is one individual bear called Lenore. So I've been monitoring her for about 13 years now. She's getting an old lady. She's coming towards the end of her life. But because we have this long-term study, we've been able to track these individuals over time and look at how they change, both within seasons, but also between years as well. So you can see by looking at these images that she looks very different at different times of year and across different years. So from the top left, where she's got like a long winter coat still, to the bottom left, where she's molted that winter coat, that's probably in the summertime. And then to the right of that, you've got when she started to regrow a winter coat, and it's much darker than what it looks in the springtime. So bears change a lot. It's very difficult to recognize individuals sometimes if you don't have prior experience, either with that population of bears or for the species more generally. So this is kind of what attracted me to the idea of our project, the BearID Project, was, are we able to develop like an automated system that is able to recognize individual bears? Because most people can't go out to the field and watch these bears every day for 6 months. I'm lucky that I work in an area with a fairly high density population of bears, but some bears obviously live at much lower densities where it isn't possible to directly view them every day. And I realized over time, watching these bears, looking at the camera trap footage, that I was able to learn how they change. However, if somebody kind of came in and saw a picture of her in the spring and then in the fall, or even in the summer, they might think that these were different individuals. So I learned that actually by like kind of tracking these changes over time, I can teach myself how to recognize them. So I thought if we can kind of incorporate all of that variation through camera trap imagery, maybe we can train a deep learning machine model to do that as well. So why is this important? Well, if we combine BearID with camera traps, it can help us to answer some questions and look at things like population estimates, which at the moment aren't possible with camera traps for bears. Study movements, look at behaviour, and then also to look at human-wildlife conflicts, so human-bear conflict issues as well. So some management focuses as well. So more specifically for this part of the project, we're wanting to track individual bears across space and time using camera trap images. And why are we doing that? Well, we want to provide data to support evidence-based decision-making, and create this tool that can be used by other researchers for grizzly bears and hopefully other bear species as well. So our data is collected in Denat-Dawit-La traditional territory in coastal British Columbia. And we used 4 years of data, 2018 to 2021. which gave us just over 11,000 camera trap days. So camera trap day is a camera up for one day. So you kind of pull all that together. And we use 20-second videos and pretty high resolution. You can get 4K camera traps now, but we haven't used them, but most of them are 1080p. And we gathered a data set of 40 individual bears that we had high confidence that we could recognize and that we'd been observing over multiple times. So we don't automatically give bears kind of like a number if they're only seen once. We have to see them for a certain number of times before we actually like categorize them as an individual. So we've documented more individuals than this, but we wanted to just like create this like core database for this project. And we're only including bears over 2 years of age because they just change so much in those really, really early years. We do all the labeling ourselves so far. We're doing species, age, class, sex, individual ID, and whether we saw a face in the video, a yes or no. So that's just basically if we saw both eyes at any point in the video. And I should have said before, we concentrated on faces because, as I showed you, there's so much variation going on the whole body of the bear that we think that the face is probably the most stable feature over time. But obviously going down to individual ID takes so long to do manually. You know, we can't just, obviously a grizzly bear is pretty instantly recognizable, but these individuals takes a lot of comparison between. So that just adds on to the time that it takes to label. So hopefully this automated system can help with that too. So we did some pre-processing of the camera trap data using a previously developed face detector, bear face detector by our research group. So that takes in the image and it locates the face and these facial landmarks, and then it produces a smaller image of the cropped face, which is just 150 by 150 pixels. So this gave me the ideal small image size to work with for the actual classification part. So here's just an example of some of those what we call face chips, these tiny little bear faces from the camera trap videos, and it kind of shows a nice example of some of the different facial poses, different resolutions, day and night. So yeah, so you can see some of this data is a little bit kind of noisy, although bears tend to like move in through fairly open areas. They use trails, they use roads, so that's kind of nice for us camera trapping. They're kind of like places with a good line of sight, so that's kind of that helps us a little bit. So let's look at the data then. So we did some data subsampling because of all of the videos that we collected over those 4 years. We had almost 3,000 videos where a face was detected and we could recognize the individual. And so by splitting that up by frame of where those faces were detected, we had over 400,000 face chips. So it was just way too much data to work with during the summer school. So we came up with different kind of partitions and some subsamples for our data so that it would be a little bit more manageable. And because of this like heavy imbalance, so obviously you can see some individuals were detected and seen much more than others. We've got this really long tail. So we wanted to try and balance that out a little bit more as well. So we did things like we created like a maximum and a minimum threshold of images per ID to be included. And we did that by looking at like the average videos per bear and the average chips per bear, just to kind of like come up with some, you know, like a metric to go by. And we had in the different partitions, different thresholds as well. So at the moment for our prototype, we've only tried the 10% subsample and the minimum of 100 images and the maximum of 230. But we also have another data set that includes like the max of like 400 as well. So it'd be interesting to play around going forward with like different thresholds and see how that affects performance. And then we wanted, because we knew there was so much variation contained within these images, we wanted to try and like select images in a way that kind of accounted for that variation. So we did a thing where we like looked at images per bear, per year, per season. And then we also like clustered to try and get some like independence. If a single bear was seen on a camera trap within an hour, it was like clustered together. And we made sure that those clusters were not split across the test train and the val. So we were like, we don't want to have any like data leakage because we don't want images that are too similar to be split across the different sets. So that gave us a data set for this project, for the summer school, for just over 7,000 chips, so much more manageable. And we converted that to CodeFlow.json so that we could have like a standardized database to work with. So we're using the Pi model, so the pose invariant embeddings, which is adapted from FaceNet, and the code base that we're using is the Wabia Pi version 2, developed by Wildbook While Me by Jason's group. It uses a ResNet 50 architecture, and it uses this triplet loss function. So the way that works is it takes an image, an anchor image of one individual, and then it has matching and non-matching pairs. So the matching pair would be a different image of that same individual, and then an image of a different individual too. So it learns to recognize and tries to push together in this like embedding space images that are from the same individual closer together, and images from different individuals further apart. And that's how it kind of does the clustering, which it uses eventually for classification. So we ran some different experiments using different epochs, whichever you prefer, different batch numbers, different learning rates. And we played around with some transformations as well, using the baseline that came with Pi, which was resize random crop and random bean, I think it's pronounced, fine. And then we added in some different transformations, like kind of a bias process, to see how that affected the performance. We did some of those augmentations just to show you visually. This is just a printout that Pi gives you of the first batch of images. So this was the baseline transformation. And then we added blur, so you can see this image here is a little bit more blurry than this one. Might be difficult to see, but it's the same set of images, so you can just compare across. Then we added grayscale by itself, because obviously some individuals may be more likely to be seen at night, so we didn't want to bias by images that were in color and red. And then the last one that we did as well was we kind of combined all of these together, and we added a color jitter. And I think the color jitter probably is a good thing to have, because if you can see here, some of these images become quite overexposed with the color jitter, and that's quite typical of camera trap images. So daytime, if you've got like the sun shining down on the bear's face, and then at nighttime if the bear's like really close to the camera. So I think that even though this may be making the work, the job a bit more difficult, I think that this is a better representation of the different kinds of camera trap images that we can collect across images. So then we moved on to some initial evaluation. So we looked at mean average precision and ranking. So rank 1, 5. So rank 5 being that the correct individual was in the top 5 predicted bears. And you can see we were getting some really good results. We were like, okay, this is really promising. This sounds great. Like, we're learning, we're continuing to learn. The gray line there was the last model that we ran using this approach. And it was continuing to learn. So we're like, okay, what can we do next to improve things? But all the time, I was thinking, this feels a bit too good to be true. Like 90% ranked 1 with camera trap images of bears that change so much. And this was getting a better result than when in the past when we've trained models on like handheld photos of bears, which are generally like higher resolution and less noisy and no nighttime footage. So we were like, can we really trust this? What do we need to do? So anyway, so I dug into the actual data set itself. I was checking things, making sure we didn't have any overlap, like, was our data set correct in the way we'd split it and everything? We couldn't find any problems. So Sarah and I sat down and we started to think about sequences, and we were like, oh, well, this is a good result. Maybe we should look at sequences and as like a second step of validation and look within sequences and look at performance. And then that's when we realized that actually the code was not, and the model was not behaving as we expected. So just to explain, you can see these images along the top here. These are from a single sequence. So because we have videos, we have frames, so it is a sequence of images. And these videos in the daytime are 60 frames per second. So even though we've subsetted with our like 10%, we can still get videos where the bear looks pretty much identical to the previous frame. So we made sure that these clusters were either in the train, val, or test. What we hadn't considered was that this triplet loss function, it doesn't account for sequences, basically. It was kind of like comparing two images, so just showed here for demonstration, that could have been from the same sequence, comparing them to one another and saying, okay, yeah, this is a really, really easy positive. And then perhaps comparing to an image like this, which is obviously like drastically different. So because we didn't account for we didn't have like a sequence ID, because we didn't think we really needed it, because we knew we were separating those like 1 hour clusters, but we didn't kind of pull that together with how the code was actually working, what was actually going on in this function. And similarly with the evaluation as well, we found out that in evaluation, these pairs may have been compared to pairs of images within the same sequence. So luckily we have the sequence ID, so we had to kind of like go back and like pull it out and add it. But basically what we think is what a lot of what we're actually learning in those results is just like being really good at recognizing sequences of bears, not being good at recognizing or not learning specific features that are useful for distinguishing between individual bears. So yeah, we thought this was just kind of a bit of a lesson learned, and I think it was a lesson learned for me in terms of like, I feel like I know my own data really well, but I didn't know the code base well enough to really understand exactly what it was doing. And that gave us, a result of like 90%. And it made me think if we hadn't have found that out, then we could have gone ahead and published this research and thought we've got 90% accuracy, like rank 1. And it's not until you like actually dig into the code. So I think for me going forward, that's going to be a huge part as well. We've talked a lot about making sure like your actual data sets are representative, but also I think for us ecologists, we have to dig into the papers and the code itself and make sure we're fully aware of how it's using our data and whether our data is relevant and representative to be used in the way that the code was written. So this all happened in the last couple of days. So it's been pretty stressful. And yeah, thanks to Jason for staying up last night till like 11 o'clock at night with me trying to figure this code out. We only got the code running and corrected towards the end of yesterday. But what we've done to try and is basically to try and adjust how the data loader selects the images that it uses to compare. So we went back, we pulled out sequence IDs. And so just for example, along the top, we have like an example of like 17 images, 18, and then what we didn't used to have was any of the sequence information. So we didn't really know the relationships necessarily, or we weren't accounting for it between the images. So what we've done now is we're making the data loader select one image, a random image per batch per sequence, so that we know the images from the same sequences aren't going to be compared to one another. So you can see in the blue how like batch 1 would like select just randomly within each sequence, and then within the same epoch, like batch 2 would select a different combination of images, and then that would iterate each time with a different epoch. It would be different images. So Jason was called like stochastic batch sampling or stratified stratified random kind of sampling within that. So yeah, and then also for the validation set, we'll only now pull one image per sequence. So again, we're just working on this right now and trying to kind of like get something working, and we'll go further to see if this is necessarily the best approach. But it gave us something to work with, and very, very preliminary model. Like I just saw this at like 6:30 this morning, so I just had time to like take a quick grab from TensorBoard. we tried to run for 2,000 epics. It only reached 1,975 before it filled up the disk space. I don't know why, because we moved it to the volume and everything. So anyway, it was just something to show that potential, and this was just the last printout because I couldn't look at the best model, but we're learning, so we're still getting some potentially, encouraging results, and we'll have to dig into those results. And this was the model we used all of those same transformations, so it's kind of like the most difficult of the previous versions that we'd created as well. So yeah,
a rank 1 of 75% is still very respectable.
Yeah,
I think that's really exciting, and now you really know that it's not. So the way that metric was working is it was taking the validation set and then verifying the cluster tightness within each individual. So it's saying of all the individuals, with the same ID, are they very close to each other in this embedding space? But if you're comparing the validation set to itself and there are sequences, it can get very good results by just clustering the sequences together. But so now here, for each run of validation, it only gets one image per sequence. So it has to be able to put the same individuals from different sequences together. So that I think is really exciting.
Yeah. So this is encouraging. And this is more what I was thinking we were going to get, to be honest, just based on some of the work that we've done in the past. This is more representative of what I was expecting. So next steps then, we've kind of gone, we made progress and we've gone back. So we kind of need to move forward again. So checking the sampling design, just making sure all the code base is as intended. Train some more models and maybe tune some more hyperparameters. Poke the model and also look into images and sequences of good performance, bad performance. And also then come back to, are we making the best use of this sequence data? And maybe we could incorporate sequences in a slightly different way. Just spend some more time considering it rather than it kind of trying to kind of do something that avoids the additional kind of information that we have for sequences. So I don't know, but yeah, just think a little bit more about it because there's something in the past that I didn't really consider. I don't think
and also how to use it.
Yeah.
Right, because it's actually useful if you get a bunch of examples. Now you can after predicting, now we can try to combine the results across all the different images of the same bear. Maybe some of them are easier than others.
Yeah, absolutely. And it makes me think as well within that, if someone shows me an image of a bear and goes, do you know who this bear is? I'll say, do you have any other images? So I can look at it from a slightly different pose. And there might be one where I'm it's that bear. So yeah, if you can cluster all of that together, then it's just going to give more information. So yeah, hopefully that will be a good approach going forward. And then just a few other general considerations and things, just learning more during these last few weeks about human in the loop approach. And I think I came to this from a very much everything can be automated perspective, and just learning about actually the realistic expectations of what we're trying to develop and how it can help people and how people can help the models as well. And then also, I guess I didn't really realize how kind of novel and different doing BearID with camera traps with video sequences is. So that's kind of exciting if we can get this to work. Sarah and Jason were saying it could be really cool paper. And also just, maybe set some kind of maybe a bit of a blueprint for other projects like this as well. And then just wanted to also mention about all these new skills that I feel I've learned these last few weeks. I'd never used command line before, really, not really used GitHub in this way or code bases, hadn't worked with VMs, hadn't really done any cloud computing, tiny bit of Python, but I definitely know not enough now. So that's something, it's definitely a work in progress. That's something that I'm gonna work on, and I think maybe it's held me back a little bit, but if I can kind of get beyond that going forward, I'll be able to do some of that digging into the code base myself and just kind of understand things a little bit further. So yeah, thanks so much to all of the instructors, TAs, organizers, and thanks to my supporters and funders for allowing me to be here. Thank you.
So when I am so impressed by the data that you're able to like learn how to recognize the faces to even label the data. I'm curious if you've heard in your field if somebody has done gait analysis with the video sequences.
Yeah, no, I don't think so, but I've always wondered about gaits analysis. So most of the cameras we kind of like orientate like diagonally, but the last few years we've put a few that are kind of like perpendicular to like a trail, just to like see maybe going forward if we could look at body shape for doing maybe age or sex. Sexes especially, I think you could do like from body shape of bears. But also, yeah, especially mature bears have distinguishing gaits, I think, from just like viewing them. But as far as I know, I don't know if anybody's done that for like large mammals or in natural settings yet. I'm not sure, but it's definitely something that I was kind of interested in. I don't know if anybody knows if anybody's.
We've been looking at it a little bit for our elephants.
Yeah.
But I think like absolutely it just hasn't been done yet.
Maybe like deep lab cut in the field or something.
Yeah, definitely.
But something like deep lab cut, you know, where you can build this very lightweight representation of like the changes in gait might be like a really nice off-the-shelf way to get something that then you can try to learn from.
Yeah.
So you don't have to start from scratch trying to do gait analysis, for example.
Yeah.
There's been a ton of work on human gait analysis for like healthcare. So like how do you sort of help people recognize like where their walking patterns are like potentially causing overuse injuries. And they're doing gait analysis for horse healthcare with deep lab cut, right? They're trying to recognize lameness in horses. And so you could maybe take the ways that they're understanding gait, like those representations, and then try to figure out the idea.
Awesome.
Yeah.
That's cool. Frank?
Melanie, super cool project. I'm just curious as to like the scale or like population we're dealing with. Are we talking like dozens of bears or like hundreds?
Yeah, so this data set was only trained on 40 individuals because we just wanted, like, it was 4 years of data, but 40 individuals. We wanted like just like a nice, clean, closed set to look at performance, like on this scale. I mean, this is a high density area, but it's only one river system, and we're working with Indigenous guardians to like expand the camera trap network. So we already have that camera trap data. So I should have probably put it in these slides, but one of the things I want to do is like, we know that these bears from this small clustered area move. So they, you know, go to different river systems. So like, even though we've only trained it on these 40 bears, it'd be really interesting to see where they occur in this like broader context, because they have pretty large home ranges. So I think, and then from there, obviously like expanding, we've talked about doing like an open set approach where we're looking at identifying like new individuals that weren't contained in the training set. That's where I think a lot of the applications to research would really come in, especially things like population estimates, market capture, where we need to be able to distinguish between new bears and ones we've previously seen. So that's an exciting thing, and kind of like Pi allows for that, and the way it does these like comparisons and clustering. So that's something that we'll be looking at going forward. And another thing that I learned during this school is like, I didn't think that it was realistic to think about open set. That's why we kind of like previously focused on closed set. So just learning some of these new techniques for myself will kind of like expand where we go next, I think.
Awesome. Really cool.
Ethan.
So I'm curious, like, if you and your group have thought about like how this might be able to generalize to other areas. Like I know, for example, in Montana, lots of like land managers would be super interested in stuff like this to try and minimize bear-human conflict.
Yeah.
So I'd be curious, like, if there was some unsupervised way to like take your model and put it in a new place and have it like sort of identify unique bears from scratch. I'd imagine that's a very different thing, but like this kind of thing would be super useful. I feel like.
Yeah, and that would be more like that open set approach where you're getting new images of individuals that may not have been contained in the training set. I think what's going to be important for bears, for grizzlies, is to make sure that if we want to do that, we have a representative sample in the training set of bears from those areas, not necessarily specific individuals, but as you know, grizzlies in different areas look pretty different. So like BC bears look quite different from Alaskan bears, look quite different from bears in Montana, just in terms of like their size. Coastal bears are bigger, but also like they maintain a longer like coat generally in the interior than they do on the coast. So we do have some camera trap data from interior BC that we want to try and build in and look at like differences between to try and see how applicable this would be for other areas too. So
yeah. Anytime you have an unsupervised question, the first thing you need is a test set with supervision. So yes, but you'd have to work in Montana to basically build the thing that's going to tell you if you're doing it right.
Yeah.
And then once you have that, then you can try all sorts of stuff.
And I think like you said about managers being interested in this, I think from some of the stuff we've done in the past, there's so much interest. Like there really is so much interest in how individual ID of species can be used for management, like problem bear issues, especially. So I think that's like encouraging that people want to use it. But it's also like these things like take time, right, to develop. So it's like, you know, how long is that going to take to get to that stage? I'm not sure yet.
One more question and then, Natalie.
I'm curious if you could somehow use the misidentification of individuals to like correlate with the status of the individuals. So like you mentioned how much they change throughout the season, and like if you get a different probability that it's this individual throughout the season, whether it like correlates with a specific, you know, health status.
Yeah, absolutely. And there are some bears that we like only see at certain times of the year as well. So I have wondered about that. And also other things like when we're talking to Tanya about like relatedness, I think like certain individuals are more difficult to tell apart like than others. So it'll be interesting. I think what I really want to do is do some of that digging into like performance on images, individuals and sequences, and just really try and look at like, I know a lot about these individual bears, so like how can I learn more about the model and how it's working based on like some of these different variables.
Yeah, it's really interesting.
Thank you.